Cell lines in continuous culture are prone to genetic drift. Finite cell lines are fated for senescence, and all cell cultures are susceptible to microbial contamination, and even the best run laboratories can experience equipment failure. Because an established cell line is valuable, you have to make sure you expand and freeze down. As soon as you have a small surplus of cells, you want to make sure that you subculture and you freeze it down as a seed stock. So for example, the first time that we freeze down, we might have like 4 vials. So 3 of those we can continue expanding, and those will be our working stocks, but one of those will have like a passage number 2. It'll just be very low passage. So that one we'll go ahead and keep it as our seed stock. This way, when we're finished with our working stocks, we always have the seed stock that we can re-expand. So our cryoprotective agent is going to be DMSO. That's dimethyl sulfoxide. Cryoprotective agents reduce the freezing point of the medium and also allow a slower cooling rate, greatly reducing the risk of ice crystal formation, which can damage cells and cause cell death. So yeah, if we just freeze cells without a cryopreservation agent like DMSO, then because there's water in the media, it'll crystallize and then your cells die. So DMSO protects them from dying in that way by reducing the freezing point of the media. Okay, so the freezing protocol will come from the company that sells you the cells. And the freezing protocols, there's like a basic one and then there's modified ones depending on the cell type. But basically you want to freeze the cells by reducing the temperature by approximately 1 degree Celsius per minute. So we use a controlled rate cryo freezer or cryo freezing container. So we have a Mr. Frosty. We've got Mr. Frosties in the lab, and that will allow the cryo vials to be frozen down slowly at a rate of 1 degree Celsius per minute, which is exactly what we need. You have to know that information for your quiz. And the frozen cells should at least be stored at -70. I usually store them at -80, and then from there we can transfer them to liquid nitrogen. So I'm not going to want anybody touching the liquid nitrogen tank. That stuff is kind of heavy and you have to wear safety protective equipment, or else if the liquid nitrogen splashes and it gets into your eyes, you can go blind. So I have to wear a face shield. So the normal freezing media that we make is 10% DMSO, 40% FBS, and 50% normal base media, so DMEM or any other media that's like a base media. So remember that we're going to culture cells in the log phase. Okay, and so we're going to go over the protocol for freezing cells in person, and I will be recording it, so I'll upload the video for you guys to practice. Thawing frozen cells. The thawing protocol is stressful to frozen cells, and using good techniques and working quickly ensures that a high proportion of cells survive the procedure. As with other cell culture procedures, we recommend that you closely follow the instructions. Thaw frozen cells rapidly. So remember, we want to freeze cells slowly at 1 degree Celsius per minute, but we want to thaw cells rapidly in less than a minute in a 37 degrees Celsius water bath or a bead bath. Dilute the thawed cells slowly using pre-warmed medium. Plate them in high density to optimize recovery. Always use proper aseptic technique. Wear your PPE. And then some freezing media contains DMSO, which is known to facilitate the entry of organic molecules into tissue. Handle reagents containing DMSO using equipment and practices. So basically, we usually remove the DMSO, but for some cell lines, you're not supposed to. You're supposed to just seed because it's actually less harmful. We have to go based off of what the company recommends. One thing to note is that when we thaw and we finally transfer our 1 ml of frozen cells in their media, and we transfer it and dilute it into their regular warmed media, we do this in a drop-wise fashion. That's very important. Types of transfection. We're not going to be performing transfections in this lab, but we'll just go over things very briefly. So a transfection is when you add nucleic acids into a cell, nucleic acids that normally aren't there, and you have transient transfections, and then we have stable transfections. Transient are just for a little while, and stable remain. So in a transient transfection, the introduced nucleic acids are only there for a little bit, and they are not integrated into the genome. So people use these transient transfections to make a high level of expressed protein within that period of time. Once you transfect transiently, you definitely want to use your cells within 24 to 96 hours. A stable transfection is when foreign DNA or nucleic acids is either integrated into the cellular genome or maintained as an episomal plasmid. So this is the maintenance of long-term exogenous DNA in the transfected cell and its progeny. So it is passed from generation to generation. So normally the efficiency isn't that high for a stable transfection. It says that you normally get a single or a few copies of the exogenous DNA that is integrated into the genome. So for this reason, the expression level of stably transfected genes tends to be lower than that of transiently transfected genes. You can also choose a transfection strategy. We're not going to go through this because, like I said, we're not doing it, but if you ever have to, this is a good resource for you to go look into. Okay, here we're just going to compare the transient versus the stable transfection. So for the transient, the translated DNA is not integrated into the genome, but it remains in the nucleus. For the stable transfection, the transfected DNA integrates into the genome. For the transient, the transfected genetic material is not passed on to its progeny. The genetic alteration is not permanent, but for the stable transfection, the transfected genetic material is carried stably from generation to generation, and the genetic alteration is permanent. For the transient transfection, this does not require selection, but for a stable transfection, you do require selective screening for the isolation of stable transfectants. So here they normally insert a different gene to create a different protein, and then along with that gene they can insert like the resistance to an antibiotic. And so for the selection process, what they do is they give all of the cells in the media this antibiotic, and all the cells that die, those cells are the ones that did not receive this exogenous DNA, and the only cells that remain alive are the ones that did receive the exogenous DNA. So for the transient transfection, both DNA vectors and RNA can be used, and then in the stable transfection, only DNA vectors can be used for stable transfection. RNA by itself cannot be stably introduced into cells. So in the transient transfection, you have a high copy number of the translated genetic material results in high level of protein expression. But in the stable transfection, you have a single or low copy number of the stably integrated DNA results in lower level of protein expression. So here you have cells that are typically harvested within 1 to 4 days or 24 to 96 hours. But in a stable transfection, you need at least 2 to 3 weeks of selection for the isolation of stably transfected colonies. So this takes a lot longer. And then for the transient, this is not suitable for studies using vectors. And then this one is. So we have gene delivery technologies. Again, this is for transfections, just different methods. We're not going to be focusing on any of that. Biological gene delivery methods. Those are all interesting to learn about if you ever get to work with them, but we're not going to be. Okay, here is terminology that is really common in the field that I think might be good for you to know. RNAi, this is ribonucleic acid interference. siRNA, short interfering RNA. These are usually 21 to 25 base pairs. They're double-stranded RNAs with dinucleotide 3' overhangs, and they're processed from longer double-stranded RNA by dicer in the RNAi pathway. Introduction of synthetic siRNAs can induce RNAi in mammalian cells. siRNAs can also originate from endogenous precursors. So these occur naturally in our bodies, but if we want to go in and silence a specific gene because we don't want that protein made, then we can create a silencing RNA for that specific gene, and we can just silence it, and then we won't get any production of that protein, or we'll get a lot less production of that protein. Short hairpin RNA, shRNA, also short interfering hairpin. These are used in vector-based approaches for supplying siRNA to cells for stable gene silencing. A strong polymerase III type promoter is used to drive transcription of a target sequence designed to form hairpins and loops of variable length. Okay, microRNA. Vectors that express microRNAs for RNAi. MicroRNAs are 19 to 23 nucleotides. They're single-stranded RNAs, and they originate from single-stranded precursor transcripts that are characterized by imperfectly base-paired hairpins. They function in the silencing complex that is similar but not identical to RISC. Chemically modified silencing RNA. Silencing RNA molecules that have chemical modifications. RISC, RNA-induced silencing complex. A nuclease complex composed of proteins and silencing RNA that targets and cleaves endogenous messenger RNAs complementary to the silencing RNA within the RISC complex. Off-target effects. Effects that occur when one or a few genes not specifically targeted show loss of gene function following the introduction of a silencing RNA. So you want to silence a specific gene, but when you notice that other genes that you did not intend to modify, they also show like a decrease in protein expression, those are off-target effects. So the effect may be mediated by the sense strand of an siRNA, which may initiate a loss-of-function response from an unrelated gene. Off-target effects can also occur as a secondary effect of the antisense strand of a specific silencing RNA, if it is sufficient. So you guys don't have to know any of these for the quiz. They're just, I wanted you to hear them at least once because you'll probably encounter those words again. Okay, so a little clarification. Here they're comparing silencing RNA, so they're RNAi, and then they have microRNA. So the first are synthetic. So the silencing RNAi, those are synthetic, short interfering. So you can buy them from the company or you can make them. And then microRNA are naturally occurring single-stranded RNAs. Okay, now we are in the appendix, and I wanted to show you this because if you have to ever culture your own cells, Gibco, the company, they actually update their cell culture handbook every year, and this is a really nice resource here in the back. So if you're having a problem like this one, no viable cells after thawing stock. So these are some of the potential reasons cells were stored incorrectly. I had this happen before. An undergrad accidentally used 40% DMSO instead of 10%, and all the cells died. So homemade freezer stock is not viable. So like when you froze it down, the cells were already dead. Cells were thawed incorrectly. So maybe when you thaw the cells, you want to thaw them in 60 to 90 seconds. And let's say that you go do something else and then you forget, and then they're in the bead bath for 5 minutes. Most likely everything will die. Thawing medium is not correct. Okay. Cells are too dilute. Yeah, if you only have less than 20% confluency of cells, then they'll probably die. Cells are not handled gently. You want to be very gentle with everything. Glycerol used in the freezing medium was stored in light. Oh yeah, like I said, a lot of these reagents, we want to keep them in the dark. What happens if cells grow slowly? Well, the growth medium is not correct. That's a possibility. The serum in the growth medium is of poor quality. That's possible. Cells have been passaged too many times. That's also very possible, especially for the primary cultures where you can only passage them like 5 times. If you go and passage them for the 6th time, you can notice definitely that the cells are growing so much slower. And at that point, it's time to buy new cells. You can also have cells that were allowed to grow beyond confluency. So let's say if you were supposed to passage your cells on Friday, but you went out of town and you didn't tell anybody, and you just left your cells in that same dish until Monday, and you come back and they just are covering the entire dish, those cells are probably not going to be okay. You can also have culture that is contaminated with mycoplasma. If that's the case, it takes a while, but yeah, they do grow slower usually. If you want additional training, the company offers virtual training labs, and you can find them at that website. Cells and tissue analysis. Understanding the structural and functional relationships of cells and tissues is critical to advancements in key research disciplines, including molecular biology, genetics, reproductive function, immunology, cancer, and neurobiology. Key components of cells and tissue analysis are cell viability and proliferation, cell signaling pathways, cell cycle analysis, and cell structure. We have a broad portfolio of reagents and kits for cell and tissue analysis, including Invitrogen. That's their products. Safety data sheet. Anytime you purchase something from a company, you immediately go to the safety data sheet so that you can figure out how exactly they want you to culture something. And we're done.